So let me move on now to our next speaker. I'll be introducing, as you know, mobilizing computable biomedical knowledge has many, many clinical and health applications, but also is relevant to other domains related to health, including public health, education, and research. And so it's my pleasure to invite Dr. Matthias Kretzler here to share some of his perspective on research that certainly does have implications for health of individuals and populations. I've had the pleasure of knowing Dr. Matthias Kretzler for many years, working with the National Institute of Health Rare Diseases Clinical Research Network, where I came to know him to be a successful collaborative researcher and caring nephrologist, dedicated not only to evidence generation through research and innovation, but also for knowledge dissemination and professional education in the U.S. and abroad to ensure that clinicians across the world have the best knowledge and training to benefit their patients. Dr. Kretzler has over 20 years of experience in integration of bioinformatics, molecular and clinical approaches, and has over 350 papers documenting that experience. His areas of interest include systems biology for renal diseases, including nephrotic syndrome, diabetes, hypertension, and autoimmune diseases of the kidney. He received his training from the University of Heidelberg in Germany and completed residencies in internal medicine and a fellowship in nephrology at the University of Munich. Currently, Dr. Kretzler is the Warner Lambert Park Davis Professor of Internal Medicine and Nephrology and Computational Medicine and Biomedical Informatics at the UM Medical School. He is also the Director of the Applied Systems Biology Core at the Michigan Kidney Center and co-leads the Nephrotic Syndrome Research Network, the Data Visualization Core, and the as well as the cell sequencing tissue interrogation site for the NIDDK Kidney Precision Medicine Project, and leads a number of international research networks as well. His team conducts studies integrating multi-type data in large data sets to enable the identification of molecular disease mechanisms and prospective clinical disease characterization. And his work and that of his colleagues has resulted in new disease predictors and the first successful phase 2 trial of a novel therapy in diabetic kidney disease in 2 decades. We are honored to have Dr. Kretzler join us today and share his perspective and thoughts on scalable, computable knowledge for integrated systems biology.
Thanks so much, Rachel, for the kind introduction. I'm very honored to start off the meeting with my overview of scalable computational knowledge for integrated systems biology. Here in our group at the University of Michigan in internal medicine and bioinformatics. We are at a very exciting time in our field of research in kidney disease because we are getting personal and precise with the way we are helping our patients fight their kidney disease. Along the lines of the meeting today, I want to introduce our concepts, how we go from data generation to data integration, knowledge extraction, and innovative management strategies for our patients. In kidney disease, we have a unique opportunity to have a patient-centered approach where we can capture the disease process from many angles. We routinely obtain kidney biopsies percutaneously from our patients, which not only allow us to generate histological images, but also use the tissue for a flurry of molecular profiling capabilities. With blood and urine, we have biofluids available, which allow us to monitor the disease processes non-invasively and relate those non-invasive profiles to the tissue-level information we get from the biopsies. And most recently, we were able to establish pluripotent stem cells from our study participants so that we can build kidneys on a chip for experimental validation of many of the associated findings we have. The key feature in this approach, aligned to the theme of the meeting today, is that with this multitude of information which we get available from our patients, data integration is critical so that we can extract meaningful information from our study participants that we can identify and on an individual patient level where a patient's disease resides, and can monitor that as a patient is exposed to environmental factors or treatment strategies. And we can predict outcomes as patients go through their disease journeys, particularly for those patients with poor outcomes, develop and define targeted therapies. This approach has been implemented in a series of global research networks where we establish shared data generation and data integration approaches. I will focus on two networks today, the Neptune Nephrotic Syndrome Rare Disease Network and the Kidney Precision Medicine Concept, to give you an introduction how we are implementing these strategies from data to knowledge in real life settings. My first case study is the Nephrotic Syndrome Study Network. That's an NIH, NCADS Rare Disease Clinical Research Network funded effort led here by the University of Michigan, myself here, together with a large team of investigators across North America. The philosophy is that we use an observational study where we have been following patients with these rare orphan kidney diseases for more than a decade now. We use the information from the multiscalar data integration to define functional disease classification, refine acute diagnosis and prognosis, bring that information to a molecular nephrology board, who then on an individual patient level can define targeted treatment options for our patients, following the philosophy of precision medicine for right trial, right patient at the right time. The network is active across 27 North American sites depicted here and is focusing on 4 specific rare glomerular diseases, which are diagnosed by kidney biopsies or by non-invasive biofluids. We have used our funding from the NIH over the last 12 years in the first funding cycle to establish the research infrastructure, to define the data capture instruments, both on clinical and molecular elements. The second round of funding cycle, we started to do data integration along the molecular, clinical, and histological framework to predict outcome measures and therapeutic targets. And we are in the third phase of the study now, which I will allude to in a moment, where we initiate precision medicine-based therapeutic trials in functionally defined patient cohorts. However, the key feature of that network relevant for the meeting today is that while we have established these cohorts with reasonable sizes, particularly for orphan diseases, with a multitude of different data sets, the main philosophy is that we bring all these different data domains together in the Neptune Knowledge Network, where clinical profiles, morphological analysis, including digital pathology archives, and molecular profiles are available for the global research community in these specific orphan diseases, so that the data can be integrated not only by the team here at the Data Coordinating Center in Michigan, but actually by auxiliary investigators across the world to identify, based on their specific research background in auxiliary study, predictors for response to standard of care, to identify novel therapeutic target and develop them, particularly with private partners from industry, and then bring these findings towards targeted clinical trials in our patient setting. And we are very proud to have achieved that we have now over 170 approved auxiliary study with robust public-private partnerships in place. And one of these examples I would like to give is the Neptune Match study, where we actually bringing the precision medicine concepts to our patients with kidney disease. We profile and analyze the patients along the lines I just discussed. Then we identify on an individual patient level which molecular pathways are active in a patient using non-invasive biofluids. And then we have linked this approach with the ongoing clinical trials in nephrotic syndrome, which are closely collaborating with us, where we take our patients who have very different disease mechanisms present under the same disease classification, like focal segmental glomerular sclerosis. We profile our patients according to the pathways who are currently studied in the clinical trials. And then we report the profiling information back to our study participants so that the study participant can select the trial who is targeting the presumptive pathway disease activation in his kidney specifically, so that we can bring the right patients to the right trial, including the outcome frequency in the trial, the response frequency to the trial interventions, compared to the conventional non-stratified approach, where obviously a multitude of different underlying disease mechanisms is present in a trial, So with the stratified approach, we hope to enrich the outcome frequency, reduce clinical trial time and clinical trial sites. And we are just activating the first 3 trials in these concepts as we speak today. A critical feature in the Neptune aspects are the data sharing and data mining resources we have developed. You can see here the software development team in our group at University of Michigan who has been responsible for a suite of software tools developed with specific contexts ranging from the open access NephroSeq, NephroCell instances for gene expression data and single cell analysis to the I2B2 Transmart platform for complex data integration across many different data domains. And this allows our investigators now to evaluate specific molecules in the participating study participants and link them back to the underlying clinical profiles and molecular mechanisms in the framework of our auxiliary study network. And I'm very happy to share experiences and lessons learned as we go forward with our work. The second example I would like to present is the Kidney Precision Medicine project, which was actually inspired by the Rare Disease Network, but now is targeting the 2 most common kidney diseases, acute kidney injury and chronic kidney disease. The goals of KPMP are similar to those I just described to you for Neptune, to identify new treatments for these very common kidney diseases using tissue profiling capabilities, with a specific goal to create a molecular and structural kidney tissue atlas for the research community globally to easily access in the specific context of interest to them, and then to define disease subgroups for targeted studies. Specific feature of KPMP is that we actually are led by our study participants, that we have patient engagement really as a center, as patients volunteering their kidney tissue for our studies. We felt that this is an essential part of that study. The strategy we have developed together with our study patient participants is that we bring people with kidney disease to the study. They volunteer for a research biopsy. The tissue and biosamples are shared with a central biorepository here in Michigan. We then have a flurry of different tissue interrogation technologies and sites who are generating multiscalar data from the study participants' biosamples. And then the data are stored, standardized, and validated in the data integration units, and then shared with the kidney tissue atlas. And I will focus today mainly on those 2 elements here to show you how this research network is generating a comprehensive kidney tissue atlas for the community at large. This is a complex operation as we have multiple clinical sites procuring the tissue, as we have the central hub who then is distributing these biosamples to the tissue interrogation sites. And really due to very careful planning of the activity by the NIH, we have an opportunity pool set of activities which allows us to nimbly respond to the latest technology development in the field. First example I can give you today already from this study is where we took healthy reference kidney tissue and used different technologies depicted here to generate multiscalar data sets from the tissue we have available in this setting, and then bring that together to a comprehensive mapping pipeline. And I would like to refer you to the bioarchive paper if you want to review that in detail. But just to give you the complexity and the different knowledge streams we are capturing, ranging from the procurement of the tissue by our study participating sites, due to the data generation, data integration, data analysis at the various knowledge domains present. What we have done, again, I think one of the key features you might discuss later in the meeting, we have established an ontology mapping of the KPMP data landscape using leading experts in ontology, including Oliver here from the University of Michigan, where we bring the different data domains together. so that we can map the data sets onto each other. And here you can see, for example, a multiscalar data integration map of tubular energy metabolism, a key feature how kidney diseases are manifesting itself. You can see here the 7 different data domains who were captured. They were mapped onto each other on a single cell level or single tissue level. Then we can define for each of these knowledge domains how the different aspects are captured and present in the kidney tissue. We then can bring that back to the nephron. Here you can see the tubules as they are traveling through the kidneys, and can map how different tubular segments generate energy. And for example, why specific segments in the proximal and distal tubules are particularly vulnerable to acute and chronic injuries because they have a mismatch between oxygen generation and oxygen consumption in this setting. So with this data generation engine now in place, we get a multitude of data pouring into the data visualization core led by our group here. And it's very clear that we will need to feature that information in many different aspects. We will have multiple points of entry into the atlas, depending if you come for structural, clinical, or molecular data. We will have multiple users for the atlas. And for that, we build our atlas capability in a use case-driven KPMP atlas roadmap, where we have different users of the atlas. We've formally assess their need. What does each user need from the data sets generated in KPMP, and then developed different analysis tools responsive to the new user. So our participants have a patient viewer available to them, the pathologist, a digital pathology repository, and the researchers in general, a KPMP Atlas Explorer and data repository. You can see the philosophy we used here depicted in this slide, that we started with the different data sources. We built cloud-based visualization, query and aggregation tools and analytics to facilitate our knowledge creation and dissemination. This has been a staged approach where we take the primary data and then go through a standard processing pipeline to make most effective use of the data for the community. We have different elements in place. A KPMP participation portal is live, where our study participants can access their kidney tissue and how it has been visualized by our tissue interrogation sites, which is a great asset for the patient. And we have received nearly very positive feedback, how meaningful it is for people to see what their kidney looks like, who has given them so much troubles. Our pathologists have an image viewer, which is available, web-based, dynamic across the world, where they can go in and assess what the structural lesions are present in the KPMP tissue. And then our clinicians, pathologists, researchers, and participants have the Kidney Tissue Atlas Explorer. The tool will go live in the next 4 weeks, where they can enter with specific molecular or structural context, complex data, single cell data, spatial transcriptomics, or other data elements. We have a KPMP Tissue Atlas repository linked to that, where the user then can select which data types are of interest to them, and can go into our data repository, and we have it set up in a binary fashion. One part of the repository is openly available to the community without any access restrictions. We have rigorously de-identified the data present in there to protect our study participants. But this allows easy access and entry to the data sets, which we used in the Explorer for expert domain users to download. And then we have protected data, which requires a data use agreement primarily to protect our study participants from re-identification. And with this, we have right now in the first ramping up phase of the study, 100 biopsies active. We have over 1,600 data sets already released in the repository. And over the last 6 months, we alone had over 3,500 distinct downloads of the data, and seeing the data widely used now in different communities. And the data sets, as you can see here, are quite complex, ranging from clinical variables to complex molecular mapped image analysis files. With this, we have developed a framework how investigators can interact with this kidney precision medicine project using published data, the cohort studies, the different technologies, our open and protected data. And the investigators can also access the biosamples for specific analysis and assays, which need to be brought back then to the study for further investigations by the community. And the philosophy really is to have open access as much as permissible with patient privacy concerns to the investigative community immediately after quality control of the data, so that we can indeed find a breakthrough for kidney health together with the scientific community globally. So with this, I would like to wrap up the presentation. I hope I've given you an overview with the 2 case studies, how we can define molecular disease processes in our study participants and bring them to interventional trials, which are executed by the auxiliary study communities, leveraging the knowledge in a global manner to define disease processes in kidney patients. The key feature, as I hope I was able to illustrate to you, is that we have dynamic, interactive processes to mine and bring data to the specific knowledge domains each user brings to these studies and research concepts. And I hope that over the next sessions in our meeting, we can learn more about innovative approaches, how to bring that forward. However, I will be remiss if I cannot thank the team who really made these studies possible. This is truly a team sport and was very much so during the last year and a half, where this resilient team quickly responded to the challenges, not only of kidney disease, but also of COVID and how this disease was hitting our patients with kidney disease particularly hard. So really thank you to a team who made that possible. But the biggest thanks to our patients who are allowing us to learn about their disease processes in the past, in the current and in the future, so that we can hopefully help them to fight their disease as effectively as possible. I thank you for your attention and looking forward to your questions in the discussion sessions which we are opening now.
Terrific. Thank you so much, Matthias, for a wonderful, wonderful presentation. You've really given us such a visual and a glimpse of what is incredibly complex data and in knowledge integration. So we certainly appreciate this opportunity for questions. I'll ask people to enter their questions in the chat. And I'd like to start with one on the word ontologies, which you brought up. And many in our community are active in this and recognize the role that ontologies can play. And you certainly described how you use them in terms of your integrating data and discovery. I'm wondering if you have any thoughts on cooperation and sharing with ontologies, who's building the ontologies that you're using. Do you see a need for coordination, standardization, regulation? And do you see opportunities to collaborate across other clinical disciplines with the work that you're doing?
Yes, I think this has been one of the key aspects which we have activated in the program proactively very, very early on, that the understanding is that if you move into these new knowledge domains, that it's absolutely crucial that you do that in a manner that your knowledge becomes and remains computable. And we are very fortunate with Oliver here at Michigan, with Peter Robinson from the Human Phenotype Ontology, with Melissa Handel from the OBO environment, to have very active members of the ontology community fully embedded in the Kidney Precision Medicine Project. And we're actually very happy to bring that concept to the Human Cell Atlas from the Jane Zuckerberg Initiative and to the HubMap Consortium as well, where we now have integrated efforts across these 3 networks to really map the new landscape of molecular defined cellular processes in health and disease in humans. And we are very happy, I think for one of the very first times that
lost a little. Let's see if we get the sound back. We're
Sorry,
back.
can you hear me?
We lost just that last sentence.
I'm sorry. So that no, I think, as I mentioned earlier, that it's critical for us to be proactive and integrative, and that we did a systematic outreach across the efforts in the field right now in molecular mapping of disease processes into the cellular landscape and context. And that this is an effort which needs and is formally integrated. We bring the cellular data sets to the cell ontology and really redefining with systematic efforts across many different organ systems how cellular processes can be mapped. And the human phenotype ontology effort from Peter Robinson, which is really bringing new grounds to genetic diseases initially. That's where our orphan kidney disease studies were very valuable case studies. But Peter and we feel strongly that the same principle needs to be deployed for common diseases like diabetic kidney disease or chronic kidney disease.
Yeah. Terrific. Thank you so much. We have a question from Brendan Delaney from London, I believe. So I think you're on similar time zones this evening. Brendan comments to a really excellent use case on community knowledge integration. Is there scope to add a more clinical face, especially in evolving patient-reported outcomes and phenotypes?
Mm-hmm. Yes, we actually in the Rare Disease Network have a very active PRO group. It's embedded now with the FDA together to establish patient and clinician-reported outcome for clinical trials. And we were really the first, at least in our domain, to bring frameworks of PROs really to clinical drug development, which have been utilized in phase 2 trials and now moving through phase 3 trials, and hopefully will lead to registration of the first new drug for FSGS in 2.5 decades. So this is absolutely critical. In the Rare Disease Network, where we have 12 years of effort already in the field in place, there has been a unique opportunity to do that. The Kidney Precision Medicine Project is just starting, and with a relatively small sample size for the clinical phenotypic aspects, we expect that we will probably need another 18 to 24 months to activate the PRO framework and to link the molecular features with the clinical and structural features. It's in the orphan space, it's fascinating to see how all these elements actually intersect, and also the social determinants of health, which in kidney disease you might or might not be aware that we see a significant burden in minority population, socially disadvantaged strata of our population. And that's the third aspect. We really need to map the clinical, individual, patient self-reported outcomes and social determinants together to identify the right treatment for the right patient at the right time.
Terrific. Thank you. And one final question. How do you see the infrastructure you've developed becoming more widely used? You've tapped on this a little, and you've talked about the patient engagement aspects, but I wonder if you have any general comments on.
I think there are beautiful platforms ready to use out there. I think we use the Transmart platform, which is an open source, open community platform, and now linked with the I2B2 platform quite effectively in our rare diseases. We are using it in our chronic kidney disease networks in the US, in Europe, in Southeast Asia, and are very happy, actually proud to see that the H3 Africa CKD network has now a Transmart instance fully governed by that network out of Africa, who allows African investigator, irrespectively of their physical localization, to access their own data and pursue analysis in that framework. In the molecular space, it's fascinating. There are multiple other efforts in place, both from the public and the private domain. The Jane Zuckerberg Cell by Gene framework is beautifully establishing open science in a very dynamic, interactive way. We will have our network data sets hosted there as soon as QC is completed and patient confidentiality preserved. I think what is critical in these discussions, however, is that we are aware about the real risk of patient re-identification. And we are keenly aware and working with our study participants who are actively governing us in finding that right balance between open access, but still protecting study participants and their interest in doing so.
Terrific. Thank you. And if I could, one more question came in. I would love to ask you if you could honor us with 2 more minutes. Chris Dimmick asks if you mentioned public-private partnerships. What role do these partnerships play in sustaining your knowledge platform?
Yes, this is an opportunity where we really invested a lot of time and effort together with the NIH and, our teams here at Michigan to develop an area of joint interest, to develop joint policies, and then understanding where knowledge can float all boats in the public and in the private sector. And we found that very helpful. We found very helpful to identify specific areas of needs, like we just mentioned, patient self-reported outcomes, for example, where we have what we refer to as ancillary study, meaning nested study in the overall framework to pursue specific questions. We have been very fortunate to be funded by the NIH and by patient interest groups and the Jane Zuckerberg Foundation for these larger frameworks, and usually use industry funding for specific targeted activities to supplement and complement and extend, but always stressing throughout the process that this has to be an activity where openness and data sharing is integrated, and have been very happy to see that industry really has changed a lot in understanding that these common data sharing and knowledge extraction frameworks are something where everybody, and particularly they, will benefit from as well.
Yeah, wonderful. Thank you again so much for joining us and sharing your wonderful perspective. You've truly given us such insight into research and in global perspective, really, and the role that patients across the world play in this. So we look forward to continuing to follow your work, and we hope you do the same with us at MCBK. Thank you again so much.
Thank you, Rachel. Thanks. It was great to present, and really looking forward for feedback and ways, because your effort is critical, because this is an area where clearly we understand that having a multidisciplinary approach to knowledge sharing is instrumental for us to impact as many lives as we can. Thanks so much for having me speak today.
Thank you.